So if you're watching this, you're probably in my drugs and human behavior class from Roosevelt University, but this is going to go over the nervous system, which is chapter 4 in our textbook this semester. So homeostasis can be loosely translated as staying the same, and it describes the fact that many biological factors are maintained at or near certain levels. For example, most of the biochemical reactions basic to the maintenance of life are temperature dependent. And so these reactions occur optimally at temperatures that are near 37 degrees Celsius or 98.6 degrees Fahrenheit. Because people can't live at temperatures too much above or below this level, their bodies have many mechanisms to either raise or lower the temperature. And so that's where we do things like perspiring, shivering, sometimes altering blood flow to the skin. So similar homeostatic mechanisms regulate the acidity, water content, and sodium content of the blood, glucose concentrations, or other physical and chemical factors that are important for biological functioning. Psychoactive drugs can also influence homeostasis. So, for example, alcohol inhibits the release of an antidiuretic hormone, and that can cause an increase in the excretion of urine. So neurons are the primary elements of the nervous system that are responsible for analyzing and transmitting information. We have more than 100 billion neurons that makes up the nervous system. And these can be influenced by hundreds of other neurons or the glial as well. Each neuron is contained with a single membrane that separates the inside of the neuron from the outside, and there's 4 regions: the cell body, the dendrites, the axons, and axon terminals. About 90% of the cells in the human brain are glia. Glia provides several important functions that help to ensure the survival of the organism, including providing firmness and structure to the brain, getting nutrients in the system, eliminating waste, forming myelin. The autoimmune disease multiple sclerosis results from a loss of myelin wrappings. And then another important function of glia is to create the blood-brain barrier, which is a barrier between the blood and the fluid that surrounds neurons. These semi-permeable structures protects the brain from potentially toxic chemicals circulating the blood. Many neuroscientists thought the role of glia was limited to the functions described above, but the simplistic view has been challenged because mounting evidence demonstrates that glia also have common or communication capabilities. So we know that the neurons perform the majority of information processing within the brain. So the cell body contains the processes that maintain life of the neuron, including the nucleus, and that's where the genetic material is stored for the cell, as well as the endoplasmic reticulum, where the cell's proteins are made. Extending from one end of the cell body are the branch-like projections, and that's what we refer to as dendrites. The other end contains one long tubular structure called an axon, and so dendrites are the main mechanism through which incoming signals from other neurons are received. We locate those within the membranes of the receptors, which are the specialized structures that recognize and respond to specific chemical signals. And then the axons, by contrast, specialize in transmitting the signals to other neurons. And then located at the end of the axons are these bulbous-like structures. They're called the axon terminals. And the neurotransmitters are stored in the terminals in these small round packages known as the synaptic vessels. This is just an image that shows you what a neuron looks like up close and all the different parts that we were talking about on the previous slide. And what you don't see marked on here is those synaptic vessels. And so that would be at the tips of the dendrites is where you would find the synapses. So an essential process for neural communication is the action potential. The action potential occurs as a result of the opening of ion channels, and that allows electrically charged particles called ions to move inside the cell. This change moves the cell's membranes away from its resting potential to a more positively charged voltage. And when the cell membrane is at rest, there's an uneven distribution of ions between the inside and outside of the cell. Specifically, there's more potassium ions and negatively charged organic anions on the inside of the cell. And then while there's more sodium and chloride ions on the outside of the cell. So the uneven distribution of ions and opened potassium channels are the sources of the negative resting potential across the membrane. Excuse me. And then in this state, the neuron is considered to be hyperpolarized. A sequential opening and closing of ion channels in the neuron's membrane generates the action potential. And so as the sodium channels open and the sodium ions rapidly move across the membrane into the cell, we call this depolarized. And selective blockade of the sodium channels prevents the action potential and thus disrupts communication between neurons. So selective blockade of sodium channels is the mechanism through which drugs such as cocaine or other like local anesthetics reduce pain. In the somatic nervous system, we see that the nerve cells that are on the front lines that are interacting with the external environment, we refer to these as a somatic system. The peripheral nerves carry sensory information into the central nervous system, and that can carry movement information back out. So a decision to move your leg results in activity in large cells in the motor cortex of one's brain. These cells have long axons, which extend down to the spinal motor neurons. These neurons also have long axons, which are bundled together to form nerves, which travel out directly to the muscles. The neurotransmitter at neuromuscular junctions in the somatic system is the acetylcholine, and that acts on receptors that excite the muscles. The body's internal environment is monitored and controlled by the autonomic nervous system. And this regulates the visceral or involuntary functions of the body, such as the heart rate, blood pressure. There's a lot of psychoactive drugs that have simultaneously effects on the brains and on the ANS. We can't control when our heart increases, or you get like the butterflies in your stomach, or there's a cop behind you and you think you're going to get pulled over, and you feel this kind of surge of adrenaline. That's your autonomic nervous system at play. The ANS is divided into the sympathetic and parasympathetic branches. So this is the slowing of heart rate by vagus nerve stimulation. That's an example of the parasympathetic branch. Acetylcholine is the neurotransmitter at the end of the organ. And in the sympathetic branch, norepinephrine is the neurotransmitter at the end of the organ. With the central nervous system, this consists of the brain and the spinal cord, and this is where most of the integration of information, such as learning, memory, and even coordination of activity, occurs here. So we'll talk a little bit here about dopamine and nigrostriatal. And when we look at chemical pathways that are implicated in the reward system, the reward part of the brain, the most prominent neurochemical theory of drug abuse is based on the idea that all recreational drugs, from alcohol to cocaine to marijuana, these stimulate dopamine neurons in the mesolimbic pathway. Over the past decade, though, we've got mounting contradictory evidence that's challenged this simplistic view. For example, although initial depletion of dopamine in the nucleus accumbens produces reductions in cocaine self-administration by rodents, cocaine self-administration is reinstated long before restoration of nucleus accumbens dopamine levels. And in several studies of human research participants, it shows that the drugs that block dopamine activity do not reduce euphoria produced by cocaine. And these are just a few of the findings that suggest that non-dopamine mechanisms play a role in the rewarding effects of psychoactive drugs. And the nigrostriatal, this is another important dopamine pathway that's kind of midbrain. And this is the nigrostriatal dopamine pathway. And cells from this course together past the hypothalamus and terminate in the striatum to form this pathway. Substantial loss or damage to cells along the pathway leads to Parkinson's disease. And as a result, Parkinson's disease is sometimes referred to as the dopamine deficiency disorder. According to treatment of the disorder, this consists of administering the dopamine precursor L-DOPA. Pathways containing acetylcholine arise from cell bodies in the nucleus in the lower part of the brain and project widely through the cerebral cortex. Cells in this region that are damaged in patients with Alzheimer's. And pathways arising from the locus coeruleus in the brainstem have numerous branches and project both up and down in the brain, releasing norepinephrine and influencing the level of arousal and attentiveness. Norepinephrine pathways play an important role in the initiation of food intake, although other transmitter systems are also involved in the very important and therefore complex process of controlling energy and balance and body weight. Serotonin and GABA. Serotonin contains pathways that arise from the brain and have projections both upward to the brain and downward to the spinal cord. Research on aggressiveness and impulsivity has also focused on serotonin. And if we look at GABA, this is one neurotransmitter that's not neatly organized into discrete pathways or bundles. GABA is found in most areas of the CNS, central nervous system, and exerts generalized inhibitory functions. Many sedative drugs act by enhancing inhibitory functions, interfering with normal GABA inhibition, such as with the GABA receptor blocker drug strychnine. This can lead to seizures resembling those in epilepsy patients. Glutamate, like GABA, is found throughout the brain, and nearly all neurons have receptors that are activated by it. Glutamate is often referred to as the brain's major excitatory neurotransmitter. The endocannabinoids, these are widely distributed throughout the brain, so the FDA has approved 4 medications that act on this system. And so the most recently approved one was in June of 2018, Epidiolex, and this was used to treat seizures associated with Dravet syndrome in patients that are 2 years of age and older. And then the CBD, one of the cannabinoids found in the cannabis plant, is the primary ingredient in the Epidiolex. Neurotransmitters are not the only neuroactive substances found in the drains. The neuropeptides are small protein-like substances produced in the cell body, whereas neurotransmitters are mainly produced in the axon terminal. Several neuropeptides in the brain produce effects similar to those of morphine and other drugs that are derived from opium. The term endorphin was coined in reference to the endogenous morphine-like substances. Several neuropeptides in the brain produce effects similar to those of morphine and other drugs derived from opium. The term endorphin was coined in reference to those morphine-like substances. So these substances are known to play a role in pain relief, but they're found in several places in the brain as well as they circulate through the blood, and not all of their functions are known at this time. So this just shows you the major brain structures. When looking at the brain of most mammals, and especially humans, much of what we see is the cortex, which is a layer of tissue that covers the top and the sides of the upper parts. of the brain and some areas of the cortex are known to be involved in processing visual information, other areas involved in processing auditory or somatosensory information. Relatively smaller cortical areas are involved in the control of muscles, which is the motor cortex, and large areas are also referred to as association areas. When we look at the life cycle of a neurotransmitter, the molecules are made inside the cell from which they're to be released. And so if they were just floating around everywhere in the brain, then the release of a tiny amount of a nerve ending wouldn't have much information value. The precursors from which the neurotransmitter will be made are found circulating in the blood supply and generally in the brain. A cell that's going to make a particular neurotransmitter needs to bring in the right precursor in a greater concentration that exists outside the cell. So machinery is built into that cell's membrane for active uptake of the precursor. And this just shows you the neurons use the enzymes to synthesize the neurotransmitters to dopamine and norepinephrine. When we look at step 3, many of the precursors are the amino acids, and these are derived from the proteins in the diet, and these amino acids are used in the body for many things besides just making the neurotransmitters. After the precursor molecule has been taken up into the neuron, it must be changed through one or more chemical reactions into the neurotransmitter molecule. This is what we call the synthesis. At each step in these synthetic chemical reactions, the reactions are helped along by the enzymes. After the neurotransmitter molecules have been synthesized, they're stored in a synaptic vessel near the terminal from which they're going to be released. The arrival of the action potential in the presynaptic terminals causes calcium channels to open, and so calcium enters the cells and assists the movement of the small vesicles filled with neurotransmitter toward the axon terminal membrane so that the neurotransmitter is released into the synapse. Once neurotransmitters are released into the synapse, they may bind with receptors on the membrane of the next neuron, sometimes referred to as a postsynaptic cell. Because signaling in the nervous system occurs at a high rate, once a signal has been sent in the form of a neurotransmitter release, it's important to terminate that signal so that way the next signal can also be transmitted. The neurons that release the monoamine neurotransmitters such as serotonin, dopamine, and norepinephrine, these have specific transporters that are built into their terminals, while other neurotransmitters, enzymes, and the synapses metabolize or break down the molecules. When the action potential arrives, the neurotransmitters are released into the synapse, and then they bind. And so the released neurotransmitters bind with receptors of the postsynaptic cell, and then sometimes you get the excitatory or inhibitory effects depending on the receptor. And then, like I said on the previous slide, with the metabolism, the neurotransmitters that release, such as the serotonin, dopamine, norepinephrine, have these specific transporters built into their terminals. And so the enzymes in the synapse metabolize or help to break down the molecules. Examples of drug actions. So through actions on synthesis, storage, release, the reuptake, the metabolism, drugs can alter the availability of the neurotransmitter in the synapse. Another way a drug can exert its effects is directly on the receptor. A drug can act as an antagonist by mimicking the action of the neurotransmitter and directly activating the receptor, or it can act as an antagonist by occupying the receptor and preventing the neurotransmitter from activating it. So drugs that affect existing neurochemical processes in the brain often affect behavior. And this has led to many attempts to explain normal, not drug-induced variations in behavior in terms of changes in the brain chemistry. The ancient Greek physician Hippocrates believed that behavior patterns reflected the relative balance of four humors: blood, hot and wet, resulting in a passionate nature; phlegm, cold and wet, resulting in a calm nature; Yellow bile, which is hot and dry, resulting in a bad-tempered nature, and then black bile, cold and dry, resulting in a gloomy nature. So the Chinese may do with only two basic dispositions: yin, the moon, representing the cool, passive, feminine nature, and yang, the sun, representing the warm, active, and masculine nature. The major theory guiding the treatment of clinical depression uh, proposes too little activity of the neurotransmitters that can cause depression, and too much can cause a manic state. Uh, because the rationale underlying the treatment of some psychopathologies, including depression, schizophrenia, Alzheimer's, this is based on correcting a neurochemical abnormality. So it's tempting to speculate that depressed individuals differ from normal people in terms of neurochemical levels or functioning. Drug treatments for the vast majority of psychopathologies are not cures. They only provide relief from the disease-related symptoms, indicating that much of the complexities associated with many psychopathologies have yet to be elucidated. So we don't have any single neurochemical theory of depression or schizophrenia that has yet obtained sufficient experimental support to be considered an explanation of it. The most prominent theory was proposed in the 60s when there was a few known neurotransmitters, but now we have more than 100, and so the neurochemical theory of mood has not been modified as of yet. With the brain imaging techniques, for centuries, neuroscientists have investigated the structure of the brain by removing it from the skull and dissecting it section by section. And we learned a lot from that approach. But the brain under study was already dead. And so imaging techniques have allowed us to study live functioning brains. And so we do this through MRIs as well as PET, what we call PET um, scanning as well. And with the MRIs, this is the magnetic resonance imaging. It's a technique that uses powerful magnets to determine the amount of hydrogen atoms at different locations in the body. It's become one of the most important tools for providing a detailed view of the brain. The most important merits of this technique are that it provides a high resolution of image of the brain's anatomy, and it's non-invasive as well. The PET scans involve injection of radioactively labeled chemical into the bloodstream, after which a computerized scanning device maps out the relative amounts of the chemical in various brain regions. This technique provides a direct measure of brain activity and an indirect measure of potential toxicity to specific neurons. So the most important limitation of PET is that it requires the injection of radioactive chemicals, which can limit its use in some individuals. So just some last minute thoughts here. Word of caution, let's say. Brain imaging is a very exciting technological advance that offers a glimpse into the working of the living human brain, but it doesn't come without limitations. The production of a brain image involves many assumptions and complicated statistical analysis, which are often not standardized from one laboratory or hospital to the next. A growing number of studies have combined cognitive testing and brain imaging techniques to examine differences between drug users and non-users. Several researchers have found some brain differences between the groups, but um, difference, no group differences in cognitive performance. Despite such findings, many investigators still conclude that drug users are cognitively impaired based solely on the imaging data. Such interpretations are inappropriate for two reasons. One, it's unclear whether observed brain differences are within normal range of human variability. And two, the brain differences in the majority of studies were not predictive of cognitive performance. So there's so much more that research that we need to do to really understand the brain and what's going on. Doing imaging studies, we must be careful to examine the behavior of interest so as not to draw inappropriate conclusions about the neural basis of cognition.